Namaskar. Are we live?
Namaskar. Namaskar. Yeah.
Technical problem.
Is there a problem, Ritu? Namaskar, greetings from Heartcare Foundation of India. Myself, Dr. K.K. Agarwal, welcomes you all to this roundtable meeting. This is 23rd meeting, and for Simao and for Heartcare Foundation of India and for myself, this is 822nd meeting in the last 6 months, which I have been doing almost 4 to 6 meetings a day. And today we have three agenda. One is a short agenda on Donald Trump. The second agenda is our issue is vaccine update. And the third, Dr. Madam Meenakshi wants to discuss something in the third agenda. I'll request Dr. Meenakshi Madam to chair this meeting and Dr. Mahesh Verma to co-chair this meeting. And so Dr. Mahesh Verma will sum up, Meenakshi Madam will chair this meeting. And I'll... No, this is every time we elect one person. It's my turn. You haven't chaired, therefore you have no choice because everybody has chaired meetings in the past. And I'll request Kalra to say a few words, and then we start.
Thank you, sir. I welcome all roundtable expert group meeting. Today topic vaccine update. Everybody is having eyes on vaccine development, the news, when vaccine is likely to come. And almost 192 trials or 193 vaccine trials are going on in various stages of development. Out of this 193, 42 have reached a critical phase in human trials, and 151 are still in animal phase, preclinical phase. So far, Russia, the Ministry of Health Department, has approved Sputnik 5 before even completing the phase 3 trial results. They have launched this vaccine. And Hong Kong is going to start a trial for nasal spray vaccine, which is it will be a dual vaccine for influenza and COVID-19. So far, among all these 42 candidates for vaccine evaluation, the most important various kinds of vaccines like inactivated or non-replicating viral vector or messenger RNA type of vaccines are under the process. So as per we know the vaccine development, on average it takes almost 7 years. But during this COVID crisis, very fast processes are on, and even then patient safety, how fast we can be, but we cannot compromise on the patient safety. And most likely the next 6 months, so possibly some vaccine may come in the market, and let's hope for the best. Now with these remarks, over to Dr. K.K. Agarwal.
Thank you, Dr. Kalra. Thank you, Meenakshi Madam and Dr. Mahesh Verma. We will have 3 short, one slightly longer vaccine and 2 smaller presentation. After each presentation, the chair will open it for discussion. My first thing is that I want to show you a couple of things. And the first thing is I'm teaching you certain things that if you develop this, you can go to give a lecture. This is what you can see. These are all our cases, and in the down you can see the Vice President of America, and you can see the expanded picture, clear-cut COVID left eye, and he was declared fit He was declared that he has no history of contact. He is fit to give a lecture. And you can see all cases, classical COVID positive showing left eye conjunctivitis. This is how you should wear a mask. Trump demonstrates to everybody that you should wear a mask with the nose open, and you have no difficulty in doing that. And we want to show you two more videos. I like Ritu to show the first video. These are the way you should take the mouth out, mask out. So this is doffing and donning demonstration done by the US President and the US physician. This is how you should do the doff and don. Ritu? These are two videos I want to show. And that too in public. Difficult है तो मैं करूं? So this is how you should play from the start. No, this is a picture. This is not a video. Video? Yes, this is a video. This is how you should go upstairs. And then you should stand in front of the public, call the media or the public. And then this is how you should take it out. Then you should double it, triple it, and put it in your pocket. And then you touch it again and again inside your pocket again. And then you touch your coat. And then again you will touch your coat and your shirt, everything. Then this is, you say, this is the correct technique of the, again you touch your body. So this is, he is demonstrating how to take the mask off. Then he is breathless. So touch your coat, everything area you should touch. And again you touch your every area so that whatever as the viruses are there, they come in the whole body. And again you put your hand in your pocket. The second one. The second one. These are the openly in media he is demonstrating how to take the mask off. Then now his physician, he follows because he says, I'm following my physician. So this is the physician.
With C-SPAN on Twitter, Instagram, and Facebook at C-SPAN.
We take you live to to give an update.
Here comes the physician. Take the mask off, put it into his pocket. And then he starts. So that's the correct way of.
Thank you for being here.
Take it out.
24 hours, the president has continued.
Let me also say at the top,
He's
my
met
prayers
with
So these are the ways his physician taught it to his president. And the president demonstrated how to take the mask off. I'd like chair to comment or a vice chair to comment. Madam Meenakshi.
Anji, thank you very much for the opportunity, and I'm very pleased to be here today. First of all, Dr. K.K. Agarwal, hearty congratulations. It's unbelievable. 800th meeting. Oh my God. I think you said 800 plus. And commitment which you display every single day is quite unique. So well done. Second is that this is a very interesting topic you have today, Donald Trump and the COVID. Dr. Mahesh Sharma, good morning. How are you? Nice to see you here.
Yeah, very nice seeing you, ma'am.
Okay.
Lovely.
Yeah, okay. Okay. So, Dr. Verma, would you like to begin commenting on Donald Trump and then we will take it to others?
I would. In fact, I wanted to unmute myself. Yeah, it's thank you, ma'am. It's a great pleasure. I always appreciate efforts of Dr. K.K. Agarwal. That he's a superman. He's a superhuman being, super doctor, because he has really shown the way. And for the last 6-7 months, he has never rested. And whether it is doctors, students, teachers, public, all stakeholders, medical organizations, judges, everyone he has brought on fold, and you know, he has shared, and I must admire him for his great determination, zeal, passion, all he has. Regarding the pictures he showed, the only comment I would like to make is that when you are at the helm of affairs, and when
you do what you like.
Yeah, when you are powerful, you all throw all the norms and all the instructions to the wind. You know, yeah, you think that whatever you do becomes a sacrosanct. I think that is not the way we are. Basically, we are all human beings. Whatever you may be, I think you know, we should not forget that all these things, you know, basically essentials. You know, whatever we may be, everybody has to follow that. And I think it becomes a long practice when at the top, people are not only doing, but they are propagating and showing in public very boldly, in a very, you know, uninhibitive manner. So, you know, I think that gives a very wrong signal. Fortunately, it has not happened in our country. And, you know, that is why I think India is different, and we stand, you know, we stand out amongst all these nations, and we're doing very well. Thank you, Meenakshi Madam and Dr. K.K. Agarwal. I think we can take it forward now.
Yeah. Anybody else would like to comment on Dr. Donald Trump? Not only the doffing and donning of the mask, but anything else that you'd like to say about the episode of the President of the United States getting COVID?
So there are three things. One is that there was totally he broken all the rules of isolation, quarantine. His president broke the rule. He was getting a epidemic in the getting a completely like a super spreader infection in White House means no protocols were followed there. That's all. The treatment protocol, everywhere there were deficiencies. That means he is a careless president. That's all.
Chaliye. So, now would we? Yeah.
So
you know, anybody else? I'm just looking around the room. Anybody has any questions, seeking clarifications on where Donald Trump can go now? I mean, will he be? Dr. Agarwal, do you think he should be allowed to?
No, he is already 10 days will be over. After 10 days, normally you are supposed to be 10 days isolation and 4 days of quarantine.
I see.
Yesterday he said, I will not take any virtual class. I will be present physically.
Yeah.
So the next meeting will be 15th of this month. He will be openly coming. And what I'm trying to say is he's breaking all the rules and regulations of CDC, which doesn't give a good impression, because the society, the anti-mask society will proliferate with this action and say, if Trump is doing, why shouldn't we?
Yeah, that is already happening. वो तो हो चुका है. All right. Okay. So, Doctor, what is the next topic you wanted us to go to after Donald Trump?
Vaccine update. I want to give a presentation.
Oh, very good. Okay. So, Dr. Agarwal, we invite you to give a presentation on that.
I was invited recently by UNESCO last week to give a presentation on vaccine, and practically all the countries were present there. That means from Canada to everybody. It was co-associated with an American Disaster Institute. So I'm giving some of the points which I gave it as a presentation, and this includes from ABC to XYZ, from very basic to very advanced. So I thought of sharing with our people to update what vaccines we are talking about. There is a critical need for a COVID-19 vaccine because it may end the epidemic. It could be important to control future epidemics, because the mistake we did in SARS and MERS by not ending up with a vaccine. Here we may end up into a vaccine, and if a new disease comes, you will be quicker, and it will be an important contribution to the science. It is technically feasible, but there are concerns of two things: possibility of antibody-dependent enhancement, which happened in dengue, and the timeline. Now, these are some of the timelines that vaccine process started as early as January 10. Now, we need to define what the disease is to understand the vaccine. It is acute, less than 3 months, manageable, so we know the treatment. It's immunogenic as a trigger. It's thrombogenic as an effect. It's inflammatory as the cause. It's a viral. It's notifiable, therefore it is infectious. It's a disease, not a syndrome. So any vaccine should be able to prevent not only the virus entry, but it should be able to make the disease shorter from 3 months to 3 days. It should take away the immunologic characteristics of the virus, thrombogenic characteristics of the virus, inflammatory characteristics of the virus, and it should be able to prevent the disease. That is the definition of a good vaccine. Now, normally a DNA enters into the cell, it transcribes into mRNA, and mRNA produces a protein. These two processes are called transcription and translation. Here, because DNA is not there, it is a RNA virus. Coronavirus directly releases mRNA, which makes the protein in the body. So we must understand when we are using a vaccine, either we use a virus as a whole, or we use its components. So we can make a vaccine out of the virus or a vaccine out of its components. Now the components we will try to understand. Simply, we understand the components as membrane, envelope, spike, and RNA. These are the 4 basic components. And you can understand it like this. This human being is the RNA. This banyan is his membrane. This shirt is his. The shirt is the envelope, and these buttons are the spikes. Okay, so you can grossly understand a virus. So therefore, if I want to make a vaccine, I can use the RNA, make a vaccine against it, use the membrane, use the envelope, or use the spike. Most of the vaccines are using the spike portion of the RNA, or I can take everything minus the human being RNA. This is called VLP, virus-like particle. That means I'm using the banyan, the shirt, and the buttons. Without the RNA, that becomes a VLP state, virus-like particle platform. Or I can use in the shirt of this buttons, I can change the button of somebody else. This becomes a vector. Vector means you are using one person and changing the button with somebody else's. That means I'm using a vector. This is how we use a vector. Now, we must understand the structure of a virus. So the virus has ORF1A and ORF1B in its outermost coating. Then this is the spike protein. Then this is E gene, M gene, N gene. And these are non-structural proteins, 3A, 3B, 7A, 7B, 8A, 8B, 9B. These are non-structural proteins. So when I make a vaccine, I need to know, either I make a vaccine or a drug, I need to know where I am acting. And in the spike, I have RBM, which is a receptor binding domain, receptor binding and the fusion protein. These two are there. Now these are the genes of the virus, spike. Now if you see a 614 gene mutation, that is at this level. The 644 gene, which does not involve RBD and which does not involve the FV, the mutation has come where the D gene has been replaced by G gene. So all of them can be involved in formation of a vaccine or a drug. Now here also we must know that this virus enters my body through the receptor binding domain or a membrane fusion which causes fusion of the virus with my cell body. Now this membrane fusion this is what I was talking about, fusion protein. And this RBM I'm talking about, with the help it enters the receptor, the cell. So once it enters the cell, it produces the RNA, genome RNA. It produces the genome RNA. Here comes the role of RNA-dependent RNA polymerase. Now this is ORF1A and ORF1B. They make the RDRP. Now they make the RDRP, and the virus gets translated, transcripted only in the cytoplasm without entering the nucleus. Now these are the structural and non-structural proteins, and what do they cause? These are the, say, NP11, NSP16 causes proofreading. These causes RNA polymerase replicate complex. They stabilizes the structure. They counteract host innate immunity. The ORF1 causes viral replication. And these are other actions of every part of the virus, what it causes. All these slides have been given to me by Dr. Pavitra. She has done a PhD on coronavirus. And here, the whole thing again I come back to is, so when I want to make a vaccine, I can use any part of the virus and make a vaccine in the body. So at this moment, we are only working on spike. All the companies are not working on any other. So therefore, 119 vaccine technologies are too small a number. The future technologies of the drug will be, somebody will say, I will block NSP3, somebody will say, I will block NSP11, NSP16, somebody will say, I will block RBD, somebody will say, I will block fusion protein. So therefore, research is only in its preliminary stage. Because at this moment, there is a Me Too reaction. Everybody is presuming today that it is the spike protein which is receptor binding, initiate infection. So therefore, at this moment, all vaccines are working on the spike. Now this spike again has a upper area and a lower area. It has a top and a bottom. Above the top, some people are saying this area is the most important. So vaccines are using total spike protein. Some people are dividing this spike protein into 13 subcomponents and try to see which component of spike protein will become. And some people say the top portion, which is the SP, the top portion of the spike is the most antigenic. So when you say 190 vaccines are preparing, all of us are wondering that what is the need of 195 different trials? Because everybody is using a different technology. Now, this everybody knows, preclinical testing, phase 1, phase 2, phase 3. And then there are 3 terms which we want to use. One is early or limited approval. Second is approval. Third is combined approval. You are doing phase 1 and phase 2 together in one trial. These are the 3 terminologies which we will be using a lot further. Now in phase 1, at this moment, there are 28 trials. In phase 2, 14 trials, phase 3, 11 trials. Limited approval has been given to 5. No approval has been given so far. But these are the most important one. Otherwise, the number is much higher. Now, how do we want to make a vaccine? I can take a virus and use it as it is. I can inactivate the virus with alcohol, with formalin, and kill the virus and use it as a vaccine. I can take the live virus and attenuate it by repeated cultures. This is live attenuated virus. I can take the gene part of the virus only, that is RNA and DNA or its plasmids, and use it as a vaccine. I can make the vaccine artificially by a recombinant technique by combining various genes together. I can use only the spike part of the virus or the RBD part of the virus and make a vaccine. Or I can use a virus-like particle or E or M part of the virus or use a vaccine. I can use a toxin, which doesn't happen because this virus doesn't have a toxin. I can add an adjuvant. Adjuvant is like whenever you go to a movie and you see very clearly, the classical adjuvant is sugar in diarrhea. Sugar के साथ साथ sodium potassium की entry मिल जाती है. So adjuvant is which facilitates entry of the virus inside the body, and that is called as an adjuvant, which can be a sugar, polysaccharide, anything. And India is using an adjuvant to inactivated virus technique. So adjuvant can be added anywhere. It can be other viruses you use for transfer of the vaccine. And these other viruses as on today are only adenovirus, but it can be a measles virus, it can be any other virus. At the moment, research is only on adenovirus. And they are using 3 types of adenovirus: chimpanzee, gorilla, and human being. In chimpanzee, they are using OX1 virus they are using. Gorilla, they are under a experimentation trial. In human, they are using 2 human viruses, adenovirus 5 and adenovirus 26. Some people are not, then we will discuss about it. Then some people today are working on monoclonal antibodies, which Donald Trump received, which is a purified type of a plasma therapy. And then you can use a plasma, which is a passive immunization, which is you produce a plasma of an infected person, or you infect a horse and you produce antibodies in the horse and use horse serum as a passive serum. Now, these are the ways why various companies are working on that. Now, when you make a vaccine, then you have further choices, permutation, combination. Somebody will say, I will try oral. Somebody will say injection. Why not dermal? Why not intramuscular? Somebody will say nasal. Somebody will say, I'll put it on the skin. Somebody will say, I'll give you a single dose vaccine. Somebody will say two dose vaccine. And then amongst 2 doses, he will say, I'll use 2 vaccine. Both of them will have a different virus, or both of them will have a different vector. Somebody will say, I'll produce a booster vaccine. And somebody will say, I will produce a vaccine which is longer lasting vaccines. So these permutation combinations are happening in the world. And somebody now will say, my vaccine has been tried in less than 18 years. Other will say, my vaccine is tried only in 18 to 55. Somebody will come back that my vaccine is effective in 55 plus. Somebody will come with 5 and 12. And then later on, they will come back and say that my vaccine has been tried in pregnancy also. So with this, now you can understand that 190 is a very small number. Thousands of vaccines should have been. The next question comes is vaccine healthy challenge. FDA says that after you are given a vaccine, you can do a healthy vaccine challenge to see whether the vaccine is effective or not. Without allowing vaccine healthy challenge, you cannot shorten the entry of vaccine in the country. Ethical or unethical, but without allowing vaccine healthy challenge, you cannot shorten the entry of virus in the society. Variolation is like MMR parties. Variolation terminology came in India with smallpox, that you take the smallpox solution and inject it into another person and produce a natural infection. So people are talking about variolation, that you take a little bit dose and inhale a little bit dose and produce a mild illness, non-ethical. Then we have a post-vaccine human challenge, which is what I have just focused about. And now there is another term called natural exposure in the family. If a husband is positive, the wife says, I don't care, I'm not going to leave it, let me also develop it. And that's happening every day as an example in our country. Specifically, the breastfeeding mother, she says, I'm not going to leave my child, let him also suffer. Now I come back to one by one, what is the status? The most important thing is inactivated or killed vaccine. That means you take a virus, culture it, and then you can kill it by any of the product. You can kill it by formalin, by alcohol. Once the virus is killed, it is ready as a vaccine. This can be alum as in, with formalin, with alum as an adjuvant. And this is the CoronaVac, which is being done. The examples of inactivated vaccines are hepatitis A, flu injection, polio injection, and rabies. These are the most easiest way, and that's the reason ICMR has entered into inactivated or a killed vaccine. But inactivated vaccine will not give you a long-lasting immunity and may need several doses over time. That means you will require a booster shot for ongoing immunity. So when this vaccine of India comes, it may be 2 vaccines. Then again, you may say you may require a booster like a flu every year or every how many months, we don't know. That's how the inactivated or killed vaccine will go. Now remember, when we talk about inactivated, I said this is the easiest to make, and you will see 3 out of 5 approvals in the world where the countries have started using them on their people have been on inactivated or attenuated coronavirus vaccine. The phase 3, limited use, that all 3 products have been from China. 3 products have got an emergency approval and they have been given to Chinese population. Specifically to all healthcare workers and all military people have been given one of these 3 inactivated vaccine by China by using 3 different technologies and emergency approval given. So I said 5 vaccines are now available in the world. 3 of them are inactivated because this is the easiest to form. Based on that, India decided that they will launch the inactivated vaccine by 15th of August. But there was a hue and cry by the media and opposition party, and they withdrew. If India was a non-democratic country, our vaccine would have been available to the people like China did it in their countries. But now these vaccines are relatively safe. The reason being, you are using the virus, killed virus, you are not using any foreign body. So therefore, these 3 vaccines are available in the world. There are phase 2 and phase 1 examples. So you can see there are so we have the phase 2 in collaboration with ICMR, NIV, Bharat Biotech is doing it. And that's what I said, that India is also making an inactivated coronavirus. And then there are other examples, China, then another is Asian nature of Kazakhstan, and people are working on the same vaccine. Idea is the same. You have a virus, you grow it, and then you kill it by any product which makes it inactivated, and then you use it. The second is live attenuated vaccine, means the virus is live. Now, when you have a live attenuated vaccine, you have MMR, you have rotavirus, smallpox, chickenpox, yellow fever. Now, these are all live attenuated vaccines. Because they are live, they are more likely to cause more reactions, but they will also produce more antibodies. It will give you a long-lasting immunity, and only 1 or 2 doses may be required because they are something like a human snake. So let me give you a simple example. When you say inactivated vaccine means कि मरे हुए सांप को आपने किसी कॉलोनी में डाल दिया. एक सांप है, वो हो गया लाइव वैक्सीन. इनएक्टिवेटेड वैक्सीन का मतलब है मरा हुआ सांप आपने किसी के घर में डाल दिया, जो रिएक्शन होगी उसे हम इनएक्टिवेटेड वैक्सीन बोलते हैं. और लाइव एटेन्यूएटेड वैक्सीन का मतलब है कि सांप का जहर निकाल के आपने घर में डाल दिया. लाइव है लेकिन उसका जहर निकाल दिया और उसको डाल दिया, तो जो लोग डरेंगे उसको देख के वो उतने ही डरेंगे जितनी मेरी बॉडी एंटीबॉडीज प्रोड्यूस करेगी लाइव एटेन्यूएटेड वैक्सीन पे. So live attenuated means you have taken the virus, and out of that virus you have taken the photocopier machine. So what happens here is, let me come back to that slide again. And this slide shows that the virus photocopier machine happens here. Replication happens here. Now this gene which causes replication, either it is removed in an inactivated virus partially or completely, or the virus is grown on a culture repeatedly till it forgets its purpose of life. That is called That is called a live attenuated vaccine. Now, in a live attenuated vaccine, as on today, India is only working on BCG, not on coronavirus. So direct live attenuated vaccines, nobody is working at the moment. That means future is, because live attenuated vaccine requires a lot of work. It cannot be produced in a shorter period of time. Therefore, because everybody is criticizing that why shorten, because they are not using live attenuated. If live attenuated was there, it would have taken much more time to produce a vaccine. The other simplest way is to use a vector. Vector means you take another virus and another virus you take and exchange or add the corona protein into that. So either the corona protein, or you can use a part of the corona protein, which is a nano part of the spike protein, or you can use the RBM of the corona, and that RBM or corona spike protein or a part of the protein or a non-structural protein or a tip of the protein, you sew it, add it to the vector. And as I said, vectors are 3 types. That is, you have a human virus 5 and 26, and you have a vector. You are using a vector is chimpanzee vaccine, or you can use a chimpanzee virus, or you are using a virus of the gorilla. Now, what Russia is doing is, Russia is saying, I will make 2 vaccines. First vaccine on adenovirus 5, and second one on adenovirus 26. And there's our 2 doses will be, the first dose will be the same spike protein, but with adenovirus 5, and the second dose will be with adenovirus 26. China says, no, we will have 2 separate vaccines. Both the doses will be either with 5 or 26, and this becomes the permutation and combinations with that. So the Oxford vaccine, which is on the top, is CHAD, C-H-A-D, C-H means chimpanzee, A-D means adeno, SARS-CoV-2 virus. So this is OX1 virus, that is the type of chimpanzee virus they are using. Now this vaccine, vector vaccines are easy to make, technologies are valuable, but it's a third virus. You are producing a third virus in the lab because you are mixing 2 virus material and converting it into a live attenuated third virus, because these vectors are live. So this is a live attenuated third virus. So you are making a khachar out of a horse and a gadha. So therefore you are using 2 viruses making a khachar. So we don't know what will happen. And that's the reason, because of this, we saw 2 cases of transverse myelitis. That's expected. So these vectors will be very prominent. They will be easy to make. And that's the reason, if you see out of the next 2 vaccines which have been approved, 2 of them belong to viral vector vaccines. So this is the phase 3 approval, and the next is phase 3 approval for early use. The second one is AD5 and AD26, and the first one is AD adenovirus 5. So therefore, what is happening is the only way you can have a virus early has to be a viral vector, or it has to be an inactivated vaccine. Inactivated vaccine will not produce you that much positivity, but a viral vector will have chances of getting complications, as we have seen 2 cases of transverse myelitis, because you are using a foreign material. It's like 2 drugs, when combined together in the market, becomes a third drug. And then you have, these are only on record. You have different types of vaccines. These are, every company is working on this viral vector vaccines. Somebody is working and viral vector vaccine, somebody is working on gorilla, somebody is working on, so I'm not naming because every data is available here, which talks about different types of technologies. Somebody is working on a nasal spray, somebody is working on a weakened measles virus in place of adenovirus, and somebody is working on the form of a pill that let me convert it into a pill. So this is how they are working. And these are the preclinical They are using corona gene fragments. These are the lots of people who are working on vector. So you will find most people are working on either inactivated vaccine or a vector. Now, this viral vector can be replicating or a non-replicating. When I say replicating and a non-replicating means, उन्होंने adenovirus का जो replicating gene था पूरा निकाल दिया. So this gene in the body will not replicate, and there can be a replicating means you have taken the gene partially out. So therefore, this adenovirus will also replicate in my human body to some extent. And the examples of replicating is Ebola and non-replicating. No currently licensed vaccines uses non-replicating viral vaccine, but on adenovirus, 3 trials are going on, including Sputnik. So the Sputnik vaccine of Russia is non-replicating vector. Now we come back to you can use a part of the virus. You can use a part of the virus and use it as a vaccine. This part of a virus can be a protein, it can be a subprotein, it can be a capsid, it can be envelope, it can be membrane. And these are the portions which people say instead of the whole virus, let me pick up only the component which is causing the infection. And these are the examples, and some of them are conjugate vaccines. So we have HIV, hepatitis B, HPV, whooping cough, pneumococcal, meningococcal, shingles. These are all subunit vaccines. Now here again, I'm showing the same slide, showing any segment of the body, you can use it to make a vaccine. Now in subunits, most of the people are using a spike protein. Somebody is using Epivax, is working several pieces of the spike protein. Somebody has taken a piece of the virus, which is important, subunit proteins. Now these are the examples. In the subunits, you are only using the protein, no genetic material is used. Somebody says, let me convert nanoparticles. Somebody says, let me stick proteins onto microscopic particles. Somebody says, let me add an adjuvant into a viral protein and make it into a vaccine. Somebody says, I will use only the RBD part. Somebody says, let me use only the peptides. Somebody will say, let me add adjuvants into it. And there are trials going on. Somebody will say, the hepatitis B vaccine shell, let me use. Somebody will say British drug maker GSK spike protein with adjuvant, viral protein with adjuvant. This is uses species of tobacco. This is interesting. The Canada-based medical legal Philip Morris uses a species of tobacco to make vaccine. So they grow the vaccine on tobacco leaf and then they make it attenuated and use it. That's what they are doing. And this is a species of tobacco. 2 people are working on tobacco virus technology. And these are the phases, various phases. You have RBD sections too. Somebody is working on an adjuvant. Somebody is using 13 parts of the several viral proteins and they select those viral proteins. They are using 8 parts of 2 viral proteins. And this is what somebody is using a vaccine in the form of a skin patch tipped with 400 tiny needles. Somebody is working. Now we come back to the last portion, and that is genetic vaccine, where you don't use the protein, but now you use the non-protein part, and that is the genetics, where you can use a RNA or a DNA. In a DNA, you take a DNA capsid and add into it the RNA. That becomes the Zydus Cadila vaccine. Otherwise, Moderna mRNA means you are artificially making a messenger RNA by synthetically making the genes and using it. This is now this is going to be the future, but it takes time. Also, out of 3 inactivated, second is vector, easy to make. Third one is a recombinant technique, and these 2 companies will be leading, Moderna and the German company BioNTech plus Pfizer plus Chinese drug maker. Now they are working on genetic vaccines. This is going to be the future because this will take a lot of time. They may take time to enter, but these will be the best vaccines according to the literature. Then you are having a These are all genetic trials going on, mRNA trials going on, self-replicating RNA vaccines going on. Then you have DNA vaccines going on. Then you have recombinant vaccines going on, miscellaneous types of people going on. Then toxoid doesn't appear here. Some people are working on intranasal. Some people are working on universal vaccine, means you take all the strains of the virus. So China group is saying is, why only one virus? Let me take all the existing strains of the virus and make a universal vaccine. Some people are working on oral vaccine. Some people are working on more important adjuvant. Some people are working on elderly. Everybody is not working on elderly. So some people are working on elderly. Some people says no, in the elderly we will try a higher dose like influenza. And somebody says that we will use a protein and by too powerful adjuvant for the use in elderly, like shingles virus. Shingles vaccine uses a spike protein along with too powerful adjuvant to make it more powerful for the elderly. Now, but at this moment, nobody is working on the elderly. So this is important, is vaccine number 2. Should the vaccine number 2 be the same? That when I use first dose and the second dose, the first dose and the second dose should be the same virus, different viruses, same company, different companies. These are matter of debates which will come back. They will have to clarify that my vaccine, because it may be the same messenger RNA vaccine, but two companies may have different technologies. Are they going to be interchangeable is another. Somebody will say, I will use two vaccines of one company here and one company here. That means I will take inactivated vaccine on my left hand and messenger RNA vaccine on my right hand. These things are important. Then dose is important. Which dose? At this moment, low dose, high dose, moderate dose, booster dose, elderly, immunity, side effect, annual. Now, these are the things which I wanted to share with you all. This is what I shared with the people internationally. And everybody at that time thought that this information should go to all the countries. So I thought of presenting this data here.
Thank you. Thank you, Dr. Agarwal. I think it is eye-opening for many of us who have no idea, who have not much of idea of vaccine. So, do we have any other inputs or, you know, any other inputs from our valued members? Anybody wants to say something?
Can I add something to this? It was a very, very nice presentation, Dr. Agarwal. Really enjoyed it. One thing is Moderna vaccine has made it's almost become a forerunner, like you said. And it has been shown that it is good for people who are 55 plus. So now they're only waiting for the safety data. And once the safety data comes, they may start the vaccination.
Madam, you have been unmute. Can you unmute yourself?
1 minute. मेरा वीडियो भी काम नहीं कर रहा है.
आपको खोलना पड़ेगा वीडियो को.
नहीं, मैंने खोला, वो कहते हैं होस्ट है.
नहीं, आपने खोला नहीं है.
ओहो, ओके, चलो.
दिल से नहीं खोला था. यस, मैडम जो बोल रही थी आपको अनम्यूट करके बोलिए.
सॉरी. नहीं, आप मुझसे बात
नहीं, मैडम जब मुझे नाम नहीं याद आ रहा कौन बोल रही थी.
आप बोल रही थी, मैंने सुना उनको. पर
सडनली यस मैडम.
डॉक्टर
Anita Chakravarti, she is a senior micro. Yes, madam.
Connection बहुत खराब है इधर. Yeah, anything? I was just telling about the Moderna vaccine. That elderly में ये शायद first start होगा. That sounds good for all of us. And Moderna is one of the better vaccines, as you said, which has a synthetic gene and which has a nuclear protein which is encapsulated in the messenger RNA. So though it's modified, it is going to give good immunogenicity. So this, I think, is slightly ahead of the Oxford vaccine. So let's see. which one comes first?
There may be people, some people are saying we'll take 2 vaccines, some people are saying I'll they do that. So there is going to be a vaccine war in the country after a couple of months. There's going to be a vaccine war.
There'll be a vaccine war.
Professor, I have a question. Oh, sorry.
Yes, madam.
Yeah, go ahead, please.
Okay. No, I'm saying that you mentioned that Hong Kong is going into trials for the nasal spray. Is this the same nasal spray which the UK NHS and the Australian government are working upon, or is it
Dr. Kalra?
I think they're different from that. It's only Hong Kong
Achha, so nasal spray.
company. They are on this doing this trial.
I showed you that many people are working on various routes.
Various things. But my question is, there appears to be only one vaccine, which is the Russian vaccine, which is using a human, adenovirus.
No, there are many of them. Russian are the one who have introduced it in the market. There are many people working on Adeno 5 and Adeno 26.
No, I'm saying, derived from the human DNA.
Yes, not human DNA, human adenovirus.
Okay, all right.
Human adenovirus vaccine. Nobody is working on a human DNA vaccine.
Okay.
So they are basically working on human adenovirus. The plus point is easily made. The negative point is that we already have adenovirus immunity in our body. Will that vaccine cross-react? We don't know. That's the minus point. Okay. Because all of us have suffered different types of adenoviruses in our body. So human adenovirus, those people who are against it, they say human adenovirus will not work in our body because antigenicity will be low. But we don't know.
I have a question, Dr. Agarwal, from you.
Yes.
People after I think 6 months or 1 year, we will get a good vaccine. But people who are already immunocompromised, what they will do after this vaccine also? How
for those people who are immunocompromised, and that's what herd immunity comes. That built up a herd immunity so that those people can be covered automatically. So all the vaccines are harmful. So inactivated vaccine in any way you cannot use. But killed vaccine, you can use them in immunocompromised. This recombinant technique, will you be able to use? The time will tell us. Because mRNA recombinant vaccine, if it is cleared, this will be the first vaccine in the world for any disease to be introduced.
Sir, I have a question to Agarwal, sir.
Yes, sir.
Sir, is there any specific role for a phase 3 trial for vaccines when a pandemic is already there? Because can we use this vaccine among the population directly after phase 2 trials?
Yes, but that phase 3 can be your, now see, let's see the example of this. Now, what China and Russia has done, they have taken a military and a healthcare workers, and they have called this as a phase 3. They say we are giving to all military people and healthcare workers as phase 3. They can do it because they are non-democratic countries. There they can dictate. China, they can dictate. But in India, you have to follow the rules and regulations. There the public choices will come. The Supreme Court will intervene to do that. There is no. So therefore, they are cut-shorting phase 3 trial and directly giving it to the public and converting that into a phase 3 trial. See, phase 3 trial says it has to be more than 30,000 people, but it doesn't say it cannot be less than, it cannot be 3 lakh or 4 lakhs or 5 lakhs. So they are using phase 3 trial as a direct on military people and health people. And by now, it's that controversy of giving it is already 2 months old. Their results will come and they will say, take it as a phase 3 trial. In India, it's not possible unless you change the law.
But even in case of vaccines, the phase 3 trial differs from that of a trial of a drug. Because if you consider insulin, you can say that whether it is decreasing the blood glucose, we can directly measure. But in case of vaccines, only after exposure to the epidemic, it can be measured.
Yeah,
so
at the moment, they are not talking about reinfection. They are not talking about, they are only talking about whether it is producing antibodies or not. That's it. So which can be done in 14 days. So at this moment, they are not talking about protection. They are only talking about whether it is producing antibodies or not. So they are not talking about protection at all, because that takes minimum, the criteria for a protection is minimum 2 months after the phase 3 trial is over. That's the criteria, minimum 2 months. That they are not waiting here, because they are saying we are not talking about protection. We are talking about antibodies development.
Can I add something here?
Yes, madam.
They say that reports are saying that the strain, the second type of strain is the G type of strain. But they say that there's not so much of mutation. So the vaccine should take care of both the strains. So that will not be a major problem. That's what many reports are coming, and that's a good news for us.
When I showed the slide,
what I'm a little bothered about is now is the time for India to prepare where all they can stock these vaccines, whether they can use the polio infrastructure to use the vaccine, and so that it gets disseminated, because now the preparatory things are most important at the urban, rural, all those sites. And I think the epidemiologists and all have to gear up for this type of work. It's ready. Yeah, I'm over.
Madam Meenakshi is in. So so this topic is unended topic. So my purpose was to start some basics, so that when you start reading about the newspapers, you know what we are talking about, and why some people are saying that they are shortening and their vaccines are not coming. So anything which is inactivated, anything which is vector based, absolutely, these are the vaccines which are coming early, but they will not be the best. The best vaccines will come with their own usual time. The best vaccines will take their usual time with that. So I request Meenakshi madam to raise the third point, and with that the debate will be, I will like Dr. Jay Krishna from IMA also to react on that particular issue, because he is representing the IMA in our forum. Madam Meenakshi.
Hanji, all right, thank you. Doctor, I have many questions on the vaccine, but वो फिर बाद में किसी और वक्त करेंगे, because time is short.
कोई इम्पोर्टेंट है तो कर लीजिए, कोई इम्पोर्टेंट है तो कर
नहीं, मैं सिर्फ एक छोटी सी बात पूछना चाह रही हूँ. Why will I opt for an injectable vaccine if a nasal spray vaccine is available?
If nasal one will never come as the first one. It's all
you don't think that nasal?
No, I don't think so.
But you don't think the nasal spray vaccines are not anywhere near fruition? Okay. Okay. All right. Now, Doctor, I want to bring up a totally different point here, which is that the Honorable Union Health Minister, Dr. Harshvardhan, has released before the nation earlier this month Ayurveda and yoga for preventing COVID-19 infection, and also for treatment of asymptomatic COVID-19 cases and for mild COVID-19 infection. So this was done by the Union Home Minister earlier this month, that is the month of October. Now it turns out that on the 6th of October, IMA has given a, and the title of that press release is "Oses to the Union Health Minister." So, usme there is an introductory paragraph. Union Health Minister Dr. Harshvardhan has released a document prescribing such and such, etc., etc. वो सब है, and उसके बाद ये लिखा है IMA ने, science demands two things. One, reproducibility of a claim elsewhere in non-conflict situations. Two, double-blind control studies. फिर आगे जाके बोलते,
मैडम मैं आपकी फोटोग्राफ्स को हटा रहा हूँ ताकि आपकी वॉइस आए. यस.
Professor Austin, IMA, would you like to read out the 5 posers to the Union Minister, or should I read them out?
Dr. Jayakishan is there. Let him respond.
Dr. Jayakishan, yeah.
Jayakishan is
actually Indian Medical Association.
He is the legal head of IMA who drafts all the legal documents and everything from the IMA.
Very
and
good.
We also have Dr. Jayalal here.
Yeah,
Jayalal is the Senior Vice President of IMA. Both of them are, at this moment, for this meeting, he is the President of IMA, President being absent. So we can interact on this issue.
Thank you.
In this particular instance, IMA was forced to release such a press statement because previously this Ayush form of treatment was recommended for general immunity only. But subsequently, it was specifically told that this type of Ayurveda and home yoga treatment will be offered for category A patient and patients with mild symptoms. So that means they are treating this disease, treating COVID patients with such medicines. For treatment, anyway, requires evidence. A specific scientific evidence is required. There should be reproducibility and there should be trials. That was lacking in this particular instance, and that is why IMA was forced to issue such a press release.
No, very good. No, that's fine. I mean, we just want to know that, I mean, my question now is, these 5 posers, the 5 questions you have directly placed before the Union Health Minister, do you see any responses forthcoming? Because this is an old battle. This is an old battle. It's not,
till now, no response has come. Till now, no response has come. It is only few days, only 3-4 days. Till now, no responses.
Yes, Dr. Jayalal.
Sure.
Yeah, IMA is much worried on this issue because the perception which is going on with the central government is integration of the medicine and purity of the allopathy system is being questioned. So when we say all the people who have the lot of issues, when they need an allopathic medicine, and they are now trying to project as an homeopathy medicine or Ayush medicine, which has some effect, which has, there is no scientific evidence.
Yes.
And we are not against any system proclaiming their superiority.
Yes.
But IMA is only thing we are worried integrating or diluting the supremacy of, or not supremacy, I mean diluting the integrity of allopathic medicine is questioned. We are saving thousands of people who are suffering with extreme disease, with allopathic medicine, but that is not projected by the health minister. He is trying to project non-symptomatic people, and you go for Ayurveda. Then what is the fun for that? Why should those people? The government should come out and say people need no treatment. You take food. That itself is good enough. You right to have your own diet. Don't project a system or when there is no treatment is needed. And if he says, come and says for advanced disease, when you come to the Ayush, then we are ready to face that challenge. So we are much worried not only on this one single incident, but either that new education policy or the NMC which they have brought in. Everywhere they say by next 20 years, 10 years, there will not be purity allopathy medicine. It's all going to be integrated medicine. That is going to be the dispel of the glory of allopathic medicine. What is going to happen for the allopathic doctors from India in abroad? Their sense. If you say that you are not going to study only the purity of the medicine, you are only just making it the fun of mixing that mixopathy. So that is a way with a great pain and concern, and we are raising this issue to the central government. But unfortunately, as usual, the central health minister has not responded, but he cannot respond because he has no evidence or no values or no documents with him to respond to our question. This is a full forth royal right questions which we are putting for the
So, Meenakshi Madam, the basic purpose of that why we should promote scientific Ayush that goes away by these types of actions.
Yes, yes.
Because if you really want to promote Ayush as a scientific statement, then you have to follow a system of making it scientifically evident.
Yes.
So these types of premature decision by the government, that they have already prematurely given a verdict.
Yes.
It was a verdict.
Exactly.
It was not an advisory. They have given a verdict: do this, do this, do this, and do that.
Yes.
With that, now what happens here is, in the first 5 days are most important. In the first 5 days, if the case is complicated, you are ending up into a death. And in the first 5 cases, I don't think so. At the moment, Ayush department is qualified enough to diagnose those cases in the first 5 days. I don't think so. So at this moment, this may not end up in people avoiding early treatment, and they may end up into the hospitals with complications. In high-risk cases, we need anticoagulation on day 1. And there is no anticoagulation available in Ayurveda. There is no strong anti-inflammatory drugs available in Ayurveda. You don't have steroids in Ayurveda. So therefore, the IMA's point of view is justified. IMA's point of view is justified. They are not asking, they are asking a simple thing, that also take the whole department of all politicians should be using Ayurveda.
Yeah.
And then why all the top politicians of the country, why did they use remdesivir, and why did they use tocilizumab in the old case when they had sickness? And the second thing is, why do you need ICUs? Make Ayurvedic ICUs and transfer all the COVID care into that. That's what IMA has written in that statement.
So therefore,
I brought this.
Just
one second. Meenakshi Madam is the former Special Secretary Health, Government of India, and she has been instrumental in the time when SARS-1 came, and she was on the helm of affairs in the Government of India, and she is at the moment chairing one meeting, which is a type of a round table, whose job is
special interest group.
Yes, whose special interest group, and these recommendations are going to the government, and she is heading one group, and the agenda for that group is how to convince the modern medicine doctors that Ayurveda is scientific. How to convince a modern medicine doctor, that means what can be the parameters, what should be the research lines, so that we can convince the modern system of medicines that we are also scientific. That was the purpose. Now, this type of a premature statement by the ministry, who has given a verdict, the whole purpose of this committee goes away. Yes, madam.
Yes, yes. Now, I want to just say something, Dr. Agarwal, in continuation.
आपकी आवाज नहीं आ रही.
Interest group should actually make this.
मैडम की आपकी आवाज नहीं आ रही है बिल्कुल भी.
Oh, sorry. क्यों पता नहीं वो.
मैडम मैं आपको बाद में कन्वे कर दूंगा.
I would be delighted कि मेरे सेक्टर को इतनी important.
Madam has been disconnected. With that, I'll request now Professor Mahesh Verma to sum up and close the meeting.
I think, yes, Dr. Agarwal, it was, as I said earlier, that it was eye-opening today, and I think a lot of learning this time, and because as all of us, although we are healthcare workers, but we don't understand much mechanism about vaccines, and I think much of it was clarified. So if you just give me a couple of minutes, I would like to sum up and carry home messages, which is born out of this this meeting today, this round table today. Dr. K.K. Agarwal welcomed. And it is 827th meeting today. There were 3 agendas regarding Donald Trump, regarding vaccine update, and then Dr. Meenakshi Datta, Madam Meenakshi Datta, Madam had something to say. So Meenakshi Datta, Madam was chairing it. I was co-chairing it. And Dr. K.K. Kalada talked about vaccine, gave a background that 193 trials are on, 42 are critical human phase stage, 151 are animal phase stage. Russia's Sputnik has already been approved, and Hong Kong trial of nasal spray vaccine is on, dual vaccine is on, influenza and COVID, and patient safety cannot be compromised, and it is expected that a vaccine would be available in the next 6 months. And then all those interesting pictures were shown, and I think very good thing which was shown through picture was left eye conjunctivitis, very typical of COVID, and also in a sarcastic way was shown what is the right way of mask wearing and taking off. And I also said that rules are thrown to wind when you think that you can get away with it. Dr. K.K. Agarwal said that broken all the rules of CDC. I think for a country like America, it is shameful. Proved to be super spreader, careless president, deficiency galore, and wrong actions, proliferate negative practices. I think it's a very profound statement. Dr. Agarwal, very nice presentation about vaccine update, and he, as being a president of CIMAO, he was invited by UNESCO, and that is where he had made this presentation. I think there's a critical need for a vaccine to not only to end the COVID, but also to prevent any future pandemic, contribution to science, community, and it is said it is feasible. The concern is that antibody should persist through the vaccine, and the timelines that it should be available as early as possible. And as earlier, he described the disease as acute, manageable, immunogenic, thrombogenic, inflammatory, viral, notifiable disease, and not a syndrome. And why he said that? Because since it is a disease, so the vaccine is possible. Normally is the DNA enters cell and makes mRNA, and here it is, COVID is a RNA virus, so it directly forms mRNA and makes a protein into the body. For vaccine, we use viruses as a whole or as its component. The components could be envelope, membrane, spike, RNA. And if vaccine is to be made, these components can be used. And mostly using, from the components, mostly using spikes. And either we use a VRLP, that is virus-like particle without RNA, or use a vector, like a piggybacking, changing the spike. The structure of the virus, it has ORF1A, 1B. It has a spike protein, and it has many genes, M, G, N, and non-structured proteins. So for vaccine, what we need to know is where am I acting? And mostly spikes are genes of virus. So virus enters body, it clings to receptor, the ACE2 receptor, and it fuses with the membrane and produce genome RNA and structural and non-structural proteins. For vaccine, any part of the virus can be used, mostly using spikes. Objective is to block mechanism. Assumption is that it is the spikes which binds to the receptors, and different portion of the spikes are being used using different technologies. And he talked about phase 1, phase 2, phase 3. And he also said that the authorities are having early approval, and combined approval where stage 2 and stage 3 can be combined together. There is no approval so far, and there's a limited approval to about 5 organizations. Activate the virus with the alcohol or live virus, attenuate it, or and use the genes, or recombinant technique, combining virus genes together, or use virus-like particles, or has adjuvant, add something to facilitate entry into the body. He gave a very good example of sugar in diarrhea, or other virus to be used to transfer the vaccine. So like one vaccine, one virus is piggybacking other virus. An example is adeno. Some are working monoclinic antibodies like used for Trump, which is more like a purified plasma. In fact, infect the horse and use its serum. That's another way. When I make vaccine, routes can be different. It could be nasal, oral, dermal, and ways can be different. 2 vaccines, booster vaccine, combination vaccine, and trial in different groups are going on. And the most important part is vaccine healthy challenge. Whether vaccine once it's ready, whether it is effective, and it has to be allowed without using it. Post-vaccine human challenge is very important, and is also important and also ethically required. And inactivated or killed vaccine, culture the vaccine and kill it, and then ready as a vaccine. Like alum is used as adjuvant. He gave examples of polio injection, rabies, flu vaccine. Inactivated vaccine may not give long immunity. He said it is very easy to make, but it doesn't give you a good immunity. Need booster more often and easiest to make. Then another he said about inactivated or attenuated vaccine. There are different techniques. It's a very common in China. 3 of them inactivated. Based on this, India wanted to launch this inactivated attenuated vaccine on 15th August, but there was a lot of hue and cry. So inactivated coronavirus in India, China, Kazakhstan, everybody tried. Then there is a live attenuated vaccine, for example, rotavirus or measles, which will give you more of reactions, more reactions. So there'll be more antibodies, but it will give you a long-lasting immunity. India is only working on BCG and not in corona. It requires extensive work and cannot be done quickly. The problem with the live attenuated is that it needs long time. Then he talked about vector-borne vaccine. Use other viruses like piggybacking, adding corona protein and use RBM of corona. Either tip or any of the non-structured part of the protein can be used. Vectors can be 3 types: chimpanzee virus, gorilla virus, and easy to make, but it is a third virus as vectors are live. जैसे खच्चर बना दी गधे से or from horse, खच्चर out of gada from horse. Next 2 vaccine which are approved pertain to this category in our country. If early vaccine, there are 2 ways: viral vector or inactivated virus. Most companies are working on viral vector vaccine or inactivated vaccine. This coronavirus is replicating, so if gene is partially removed, it would be replicating. For example, Ebola. He said part of virus can also be used as a vaccine instead of whole virus. And he's talked about spikes, proteins, or pieces of virus. And then he talked about universal vaccine, where all the viruses, immunity against all the viruses, different strain of viruses, oral vaccine, elderly vaccine, whether different dosages should be same virus or of a different companies. There's a big debate. He talking about doses is very important, high, low, booster. And then Dr. Anita Chakravarti talked about Moderna virus. She said it's good for 50, 55 plus, but it's awaiting safety data. Dr. KK Agarwal shall be vaccine war in coming time. There'll be whole of these companies bringing different vaccines, so they'll be vying with each other for commercial availability. Meenakshi Datta ma'am, nasal spray, is it same or like Australia is working on Russian introduced human adenovirus, easily made, but we have adenovirus immunity in body, whether it will react and antigenicity will be low. That was talked about Dr. Agarwal. Then Dr. Upasana Arora for immunocompromised, whether can be used. Dr. KK Agarwal, yes. Dr. Jayakrishnan, any specific role of phase 3 trial, whether can be used directly after phase 2 on patients. Dr. KK Agarwal, in China, they have done it. They have tried it on healthcare workers. They have tried on military, cutting short of the phase 2, and they brought phase 3 directly, introducing to public. But I think in India, it may not be possible because we have to follow all the protocols. At the moment, vaccine is being talked, but production of antibodies, that is what is most important. Dr. Anita, reports say that many strains are there, but not much mutation. And we need a lot of epidemiologists. We need to gear up for all this. Dr. KK Agarwal intended sharing basics and sensitization through this presentation today, but best vaccine would take its own time. So don't think that the best vaccine is going to take long time. Meenakshi Datta, why would I opt for injection if nasal available? K.K. Agarwal nasal may not be available. Dr. Meenakshi Datta, in October, Honorable Minister introduced yoga. IMA released 5 posers, and there were basically 2 demands: reproducibility and double-blind studies. And Dr. Jayakrishnan clarified that Ayurveda is only for general immunity and select patients, and for treatment you need evidence. Meenakshi Datta, madam, whether 5 posers of Union Minister have been responded, Dr. Jayakrishnan and Dr. Jayalal, there has been no response, and IMA is worried. As homeopathic and Ayush, there is no scientific evidence. IMA is worried for diluting the credibility of allopathy, and don't promote systems if there is no evidence. We are not against any system. Integration will take away the glory of allopathy. I think very, very good statement. And there is a lot of great pain and concern amongst IMA. Dr. K.K. Agarwal, if Ayush to be treatment component, it has to be evidence-based, given a clear verdict. First 5 days are very crucial. Ayush is not competent to diagnose in the first 5 days of patients. So important to diagnose so that there are no future complications. There are no steroids. There are no strong anti-inflammatory drugs available in Ayurveda or homeopathy. So why all the top politicians are using remdesivir and tocilizumab? So I think we had a lot of healthy discussion, and we had a lot of understanding, especially the vaccine part, and how are different vaccines and what are the pros and cons and availability, dosage. I think a lot of clarity came through the presentation of Dr. K.K. Agarwal. I really like to congratulate him, and the associated doctor who got PhD. I'm not getting her name, and I think they have done a lot of work. So thank you so much, Dr. K.K. Agarwal. Wonderful. It has been a wonderful and a very, very productive roundtable webinar. Thank you.
Thank you, sir. Dr. Kalra, to end up the meeting.
I think very good as usual. This session was very, very informative, and for the benefit of all our members. And probably now we are much aware about vaccines development, how the vaccines are developed, and what are the trials going on. I think thank you very much all. I thank everybody who was attending, and again thank you all, and see you again the next week, same time.
Next week, we in the last masking meeting, we had passed a resolution that Heart Care Foundation of India will consider filing a PIL in the High Court regarding the problems faced by the mask, because most of the masks are fake. So I'll give a final presentation. Half session will be based on that, on masking. And the half session, all of you can decide, depending upon at that time what is prevalent, we will pick up. So we'll have 2 topics. First will be an upgrade for what we have done in our masking. I will give some updates on that. Thank you.
Dr. K.K. Agarwal, see some of the basic essentials which we learned today on vaccine. Is it possible to be shared? Because I'm very much interested. I got so much interested in vaccine. There are different. Yeah. Thank you.
I'll give you the slides, plus I'll give you the link. You can watch this program again and again, and I'll also give the. No problem.
Thank you very much, sir. Thank you.
Thank you, everybody. All of you are you're requested to enjoy your tea cum lunch in your respective place.
Thank you, sir. Thank you.